Welcome students. In this video, I am going to discuss about the molecular structure of DNA at chemical level because it helps you to understand how the higher order structure of these molecules influence their properties and interactions with other biomolecules like proteins, and it also helps you to understand physical and chemical properties of DNA and how it encodes genetic informations and how it passes genetic informations from one generation to the next. So let's start. DNA or deoxyribonucleic acid is a type of nucleic acid and it is linear polymer of nucleotides. Nucleotides are arranged in particular order and linked via phosphodiester bonds. Imagine this is a piece of DNA and you can see the blue boxes, that is our nucleotides, which are arranged one after another in linear fashion and joined with each other via phosphodiester bonds. All nucleotides are composed of three components. Means if you choose any of these nucleotides, you will observe there are three components: one or more phosphate groups, ribose sugar, and nitrogenous bases. Phosphate or phosphoric acid, when dissolved in water, it becomes like this, because we know that our body contains 70% water, and yes, it interacts with water molecule all the time inside the cell. Phosphate group is negatively charged and acidic in nature. So DNA is also negatively charged and acidic. When basic dye like Giemsa or ETBR is applied, it stains the chromosome or simply the DNA. The number of phosphate group present in nucleotides is variable from 1 to 3. It will be discussed later in this video. Second component is nitrogenous bases. The nitrogenous bases are planar, means all the atoms are in the same plane. It is aromatic and heterocyclic molecules. There are two families of nitrogenous bases, purines and pyrimidines. A pyrimidine composed of a six-member ring of carbon and nitrogen atoms. The members of pyrimidine family are cytosine, thymine, and uracil. Atoms in these bases are numbered from 1 to 6 and in clockwise direction. Purines are larger, with a 6-member ring fused to a 5-member ring. The purines are adenine and guanine. Atoms in these bases are numbered from 1 to 9 and in anti-clockwise direction. Remember, the number 1 position of purine and pyrimidine are not from the same position. Now I will share one simple way to remember the complex structure of those nitrogenous bases. First, draw this structure. Then write these two carbon atoms here and one double bond. Now add one keto group here. Then start writing atoms from this position. Write carbon and nitrogen in alternative position in this way. Your basic structure is ready now. Now start with uracil. Add one more keto group here. It is called uracil. When you write amino group instead of a keto group, it becomes cytosine. And in uracil, if you add one more methyl group here in this position, now uracil becomes thymine. In thymine, double bond also changed its position. Now purine. In purine, there is one 5-membered ring fused with 6-member ring. I have already told you. Again, draw two carbon atoms here in this position like pyrimidine and draw two extra double bonds here and here. Then go back to this position like pyrimidine and start writing carbon and nitrogen in alternative position like this. Carefully watch the numbering. I have finished numbering 6-member ring first and then move to the 5-member ring in this direction. Now in adenine, add one amino group here in this position And in case of guanine, change the position of amino group from here to this position and draw one keto group here in this position and now balance all atoms. I hope this will help you to remember these complex structures. Now the question is why they are called base and why in presence of these tail makes DNA acidic. Here is your answer. Pyrimidine and purine contains the amino group that are weakly basic, and the nitrogen atoms of purine and pyrimidine tend to take a proton from solution. Therefore, they are basic in nature. But phosphate group are strong acids than nitrogenous bases. That's why the overall charge of DNA is negative and acidic in nature. Now the last component of nucleotides, that is the sugar molecule. The sugar present in nucleotide is a pentose sugar, that is D-ribose. In nucleotides, pentose are in their beta form, that is a close 5-membered ring. Carefully see the atom numbering of the sugar molecule. Instead of using 1, 2, 3, 4, a prime is added in order to distinguish atoms of the sugar molecule from the nitrogenous bases. Otherwise, if I told you number 1 carbon atom, you will confused which one the carbon of sugar at number 5 position or the carbon of nitrogenous base which is also at number 5 position. The sugar molecule is change in DNA. You can see that one oxygen atom is missing in the 2' position. That's why this is called deoxyribose. The two sugar, deoxyribose and ribose, may be differentiated very easily by means of specific color reactions. Ribose reacts with orsinol in hydrochloric acid solution containing perchloride, and deoxyribose reacts with diphenylamine in acid solution. This color reaction also helps you to identify the DNA from other nucleic acids like RNA. Now let's see how these 3 molecules are attached to make one nucleotide. In 5' position of the sugar, one or more phosphate group is attached. The name of the bond is phosphodiester bond. When only one phosphate group is there, it is called adenosine monophosphate or in short AMP. When 2 phosphate groups are there, it is called adenosine diphosphate or ADP. And if 3 phosphate group is there, it is called adenosine triphosphate or ATP. The first one is called alpha, second one is called beta, and the third one is called gamma phosphate group. And if no phosphate group is there, it is called nucleoside. You can see the nomenclature is also different for nucleotide and nucleoside. Any one nitrogenous base among 4 is attached at 1' position. For purine, number 9 nitrogen atom is bonded, and for pyrimidine, number 1 nitrogen atom is bonded in this way. The name of this bond is N-beta glycosidic bond. So in nucleotide, one D-ribose is there and one or more phosphate group is attached at the 5' position and one nitrogenous base is attached at 1' position. Now check out the structure of other 3 nucleotides. This is the structure of guanine, this is the structure of cytosine, and this one is the structure of thymine. Now the question is, if I took these nucleotides and this one, are they same? The answer is no. For example, in this nucleotide, you will see 3 components, phosphate group, D-ribose, and adenine. But in this, you will see phosphate group, D-ribose, but cytosine. And therefore, nitrogenous base actually decides the name of the nucleotides. Like if adenine is present, it's called deoxyadenosine. If cytosine is there, it's called deoxycytosine. Now let's see how nucleotides are attached one after another to make linear strand of DNA. Successive nucleotides of DNA are covalently linked through phosphate group bridges, in which the 5' phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of the next nucleotide, creating a phosphodiester linkage. Thus, the covalent backbone of nucleic acid consists of alternating phosphate and pentose residue. That's why this is called sugar-phosphate backbone. And the nitrogenous bases may be regarded as side groups joined to the backbone at regular interval. Notice that only one phosphate group is here. You can write a piece of DNA in this way. Do you notice that the both ends of the DNA strands are not same? One end contains free 3' hydroxyl group that can react with other nucleotides. This end is called 3' end, and in other end one phosphate group is free. This end is called 5' end. So DNA is polar molecule, like North Pole and South Pole of the Earth. Both ends are not same chemically. Generally, DNA is written from 5' to 3' direction, and it is universally accepted notation for writing DNA structure. So if not mentioned, you have to assume that this end is 5'. So the opposite end will be 3' end. Now we will see the schematic representation of nucleic acid. DNA is represented in various ways, like this. But sometimes, for simplicity, DNA is represented in various ways, like this. Here, the phosphate groups are symbolized by P, and each deoxyribose is symbolized by a vertical line from 1' carbon atom to the top. That means the 5' carbon atom, that is at the bottom. The connecting lines between nucleotides are drawn diagonally from the middle, that is the 3' carbon atom of the deoxyribose of one nucleotide to the bottom, that is the 5' carbon of the next. Some simpler representation of these pentadeoxyribonucleotides are this, or more simply this one. So we can say all four nucleotides A, T, G and C makes the DNA. Uracil is generally absent in DNA. So all four nucleotides are arranged in different order in order to code different characters. This is called strand of DNA or DNA strand. This is also called primary structure of DNA. From this structure, one information we can get, that is the sequence of bases or their respective position in the piece of DNA. For example, the sequence of the two genes are as follows. You can see that the sequence are not same. That's why they code two different informations. So information actually stored in the sequence of bases in a piece of DNA. You can see the beauty of this molecule. Only 4 bases are arranged in 2 different ways to code two completely different information. So that's all for now. In the next video, I will discuss the secondary structure of DNA and some important discoveries that leads to the elucidation of the 2D structure of DNA in great details. If you like this video, press the like button below and make your valuable comments. And subscribe my channel for more videos like this and press the bell icon for updates. And thanks for watching.